Sticks and stones can't break my bones. Using the LRP5 V171 mutation to increase bone density in mice.
This is Team 13's presentation. I'm Anessa.
I'm Olive.
And I'm Colby.
The problem that we were researching is that there's mutation in the LRP5 gene, which has been shown to cause low bone density, which can lead to the disease of osteoporosis. The LRP5 gene, which is a co-receptor, provides instructions for the creation of the LRP5 protein that is located in the outer membrane of many cells. The LRP5 protein participates in the WNT signaling pathway, which regulates bone metabolism in humans and other mammals. To the right is the human genome, and on the 11th chromosome is where this mutation occurs. Osteoporosis is the bone disease that's been generated through this mutation, and it can cause bones to become weak and brittle. This can affect all ages, though it's most common among older individuals. It's estimated that 1 in 3 women and 1 in 5 men over the age of 50 will experience an osteoporotic fracture. And according to a 2015 study, osteoporotic fractures were responsible for more hospitalizations than heart attacks, strokes, and breast cancer all combined. Approximately 10 million Americans have osteoporosis, and 44 million have low bone density, which places them at increased risk for this disease. On the right, it shows a healthy bone and then also a bone with osteoporosis.
So for the proposed solution, there is a mutation in the LRP5 gene, which increases bone density and results in unbreakable bones. The experimental plan is to inject the mutated gene into the mice embryos, and this will be done through an engineering approach. And additionally, the mice were chosen due to their genetic similarities to humans. The expression of a new mutated gene will increase the bone density of the mice, and the modification of the LRP5 gene with this mutation could potentially increase bone density and possibly benefit humans. When considering how it benefits humans, it would increase bone strength, which could provide a potential treatment to those with osteoporosis or other degrading diseases, as stated before. It can also prevent brittle bones and decrease the likelihood of bone fractures. For the feasibility aspect of this project, a procedure of injecting the mutated gene into mice embryos is a very practical operation. Injection of genes is a common method for many researchers. There's also a vast amount of people who have osteoporosis, yet there's no cure available. And the success of this project could bring us one step closer to developing a way to treat those with the disease. For the overview of methods, it is our goal to address the low bone density by putting a mutation of the LRP5 gene into mice. We'll do this by isolating the gene by making cDNA, then transforming gene with recombinant plasmids, and after that, confirming the success of the gene transfer using DXA or sequencing the genome. For the model organism, this is the C57BL/6J mice, or mus musculus. The biological characteristics of the mice closely resemble those of humans, as stated before. They can simulate human genetic disorders such as osteoporosis due to the fact that they have a low bone density. When isolating the LRP5 gene, it is necessary to make cDNA. This will be done by isolating the mRNA from the tail of the mouse using a Gentra Purigene kit. This is a missense mutation in the mice. And we will then synthesize the cDNA from the mRNA using reverse transcriptase. And once that is done, we will make the cDNA double-stranded using either reverse transcriptase or DNA polymerase.
So in order to amplify the gene, we will perform PCR on the isolated cDNA. And 2 DNA sequencing reactions will be performed using primers that we will be obtaining from IDT. Now we identified the primers that we'll be using for this reaction by also looking at the sequence of another gene found in the liver, which is the place where we will eventually be injecting the plasmids that we make with this. So we used primers that were from that gene so that they will work in the mouse once we inject it. And then we will insert the cDNA that is copied from the PCR into plasmid vectors, as well as adding the AMP-R antibiotic resistance marker. Okay, so for this is our vector diagram. And for this, like we have our NCOL component, which is the restriction enzyme, which will be at the 615th base pair. We also have the up2 promoter. This is from uridine, which is the gene that we use to find our forward and reverse primers. So it is also found in mouse livers, which is where we will be eventually putting this gene. And then we have the LRP5 version 171, and that is our gene of interest. It's the mutated version of the gene that increases bone density as opposed to reducing it. And then we have the SALI 2025 restriction enzyme, which is at the 2025th base pair, as well as the AMP-R antibiotic resistance marker, which will allow it to grow an ampicillin, and the ORI, which is the origin of replication. And so in order to find our modified plasmids, we will grow E. coli cells that have been transformed by putting the plasmids inside them. And then we will grow them on ampicillin plates. And in order to identify the colony that has the cDNA, we will be identifying the colonies that survive after growing in the ampicillin, because the ones that we have modified are the ones that have the AMP-R antibiotic resistance marker. And ampicillin is the antibiotic that it has resistance to as a result of having the AMP-R marker. So any surviving colonies will contain the modified gene.
To create these genetically modified mice, we will inject the plasmid into mouse embryos and then implant the injected embryos into pseudo-pregnant mice. We will then test resulting offspring for the presence of the up2 promoter and LRP5 gene. Using a deceased mouse liver, we will use either PCR or DNA sequencing and the Western blot test to test for success, and then breed successfully modified mice to create more. The method we will use for confirmation to see if the gene is now encoded, we will use dual energy X-ray absorptiometry, also known as DXA, one of the machines to test bone density on modified mice, and we will also test the control mice to compare, and then we will sequence the genome. The controls that we will have during this experiment are going to be using non-modified mice, as they are the exact same strain as the one we'd modified before. The environmental factors will stay the same, as we will have the same size habitat for each groups, and the amount of exercise will be consistent. They will also both be given the same diet, and we will test mice relatively around the same age.
Okay, so for our evolutionary context of the LRP5 gene, we found that LRP5 is a member of the LDLR family, which is a low-density lipoprotein gene family, and it is responsible for cell signaling. So as shown in the diagram to the left, this is a diagram of the LDLR family and how closely they are related. LRP5, LRP6, LRP4, and LRP8 all play roles in bone regulation. So all of these are pretty closely related. And LRP5, the LRP5 gene is found in all varieties of vertebrate species. So basically it's found in any species that has bones, but because different species have different number of chromosomes, it will most likely be found on different chromosomes, or it is found on different chromosomes than it is in humans. So it's not always reliably in that same spot, but they all have the same gene. And also the ethics surrounding this experiment would be that manipulating the LRP5 gene to make bone density higher could potentially lead to unforeseen health problems, because not a lot of research has been done on this gene in particular, and especially this mutation of it. So if we were to modify the mice to have this, or potentially modify humans, there could be a potential disease related to it, similar to how osteoporosis affects your bones. So like it may be that having higher bone density could have adverse effects that we don't entirely know about, or that it could cause other issues because the gene could be related to something that we don't know. Like right now we know it's related to bone density, but it could be related to something else. And if released, the genetically modified mice could have undesired effect on the mouse populations in the surrounding ecology or the ecosystems, or like if they were to spread even further, it could have adverse effects globally. This shouldn't be an issue in this test because the mice will not be released after testing. Most of them, or maybe even all of them, will be euthanized. And so they should not be released, and they will be kept in a lab environment where they will be secure. So releasing them into the local ecosystem shouldn't be an issue.
Yeah, so for future directions, we will begin LRP5 gene editing on model organisms that have similar characteristics to humans, such as primates. And what's nice about this is that the LRP5 gene is located on the same chromosomal location as in humans. And so with that being said, and if all is successful, then we can go on to gene editing on human subjects. And by doing this, we will increase bone mass density and potentially prevent/delay osteoporosis. For an example, this could help astronauts, and it would provide stronger bones, which can increase the amount of time spent in space. Lastly, by using the LRP5 gene editing methods as a base, we could treat other genetic bone conditions such as osteogenesis imperfecta, osteopetrosis, or achondroplasia. Yeah, here are references.